We're looking at Chapter 14. Chapter 14 is meant to start giving us an introduction to some basic concepts of evolution. Now, I teach an entire course, an introductory course on evolution, and in 16 weeks it's not enough to give the field, the discipline, any due credit. We just barely gloss over things. So, in these 3 chapters, we are going to oversimplify and really gloss over what is in-depth and nuanced discipline, similar to what we've done all semester. This is meant to be a broad introductory course, and it's the same thing here with evolution. But in order to start to talk about the examples and the some of the basic concepts in evolution, we do have to have a shared vocabulary. So evolution, broadly defined, is changes in a line of descent over time. So essentially we're looking for morphological changes, those physical characteristics that change from generation to generation and change fairly significantly when we have a speciation event. So we know now that a lot of these traits, these physical morphological characteristics that we see, are based on genes, based on the DNA, and based on some of the things that are not gene-related, like epigenetics. So we're looking for the changes in the DNA, mutations, epigenetic changes that lead to physical characteristics that we can measure, those changes over time. Now, when we mention over time, time is going to be species-specific. So if I'm dealing with something like a mouse, mice can breed very quickly, and I can see changes in a line of descent in just a few generations. But if I'm dealing with something like an elephant or a blue whale, or some of those really long-lived species like redwood trees, I'm going to need a much longer time frame in order to see any changes in a line of descent. So even this concept of over time is not very static. Some other terms that we need to define or that we talk about will be things like fitness. Now, simply defined in the textbook, fitness is the ability to survive and make babies, survive and reproduce. And that definition is way, way, way oversimplified. Because for so many species, it's not just enough to make the babies. The parents, most likely the female, has to maintain their own fitness until the young are reproductively mature and can make it on their own. So when we look at species of whales, the whales' babies have to stay with the mom for a minimum of 2 to 5 years. We've got moose, where the moose babies have to stay with their mom for about 2 years. And what we have discovered from research is that if the mother dies at any point during that very, very crucial few years of life, then the young are less likely to survive. And we know this is true when we look at our own species, humans. At a minimum, parents have to maintain their fitness, caring for their children for about 8 to 10 years. That seems to be the minimum age range when children can survive without their parents. So when we look at fitness, it's not just enough to make the babies. Most species, or a lot of species out there, also require the parents to maintain their fitness until the young are reproductively mature and can leave and make it on their own. So another term that we need to have a shared common definition for is speciation. Speciation is changes in a gene pool that cause a group of individuals to no longer be able to interbreed with the original group of individuals. We talk about this term gene pool. Gene pool is all the shared genes for a species. So then if we have significant changes in the gene pool that make those individuals unable to interbreed, we most likely have a speciation event. That splitting of one species into 2 or more species that are no longer able to interbreed and make viable offspring. And we have to put that qualifier in there, viable, meaning if I mate a horse and a donkey together, yeah, they can make mule babies, and mules can have really great lives. But the mules can't keep making more mules, keep making more mules. We can get individuals from different species that mate together, but the kids may not make it. And if the kids do make it, they may not be able to make their own kids. So we don't have a true speciation event. So we do have to be kind of careful here to make sure that we qualify what that interbreeding would look like, and that we have a perpetuation of the species, that we can keep the species going over multiple generations, we have viable offspring. So two more terms that are important. When we are looking at speciation, we are looking for ancestral characteristics, those traits that are passed from one generation to the next, and more importantly, passed from one speciation event to the next. In other words, we're looking for the closeness, closely related species to have some similar traits with other species that are closely related to them. But if we have a huge separation, if we have a derived characteristic, something new, that new trait will be new to the speciation event. It will distinctly separate those individuals from the original species. So we look to see what sticks around, ancestral characteristics, even when speciation happens, and we look to see what is brand new. What does this new species have that's giving them some kind of advantage or perceived advantage over the prior species? So we have the shared definitions, the shared vocabulary, so we can start to talk about things like the image that we're seeing here, which is really huge leaps, huge jumps, as we go from life in the water, more fish-like organisms, to life on land. And we're looking for those closely related species. As each speciation event occurred, the new species would have those derived characteristics. In the case of Tiktaalik, we see the loss of that, the hind limbs, and a change in some of those front forelimb structures, allowing them to start to spend more time on land. And there's a lot of other characteristics, derived characteristics, that happened with Tiktaalik. But then there are also some ancestral characteristics, almost throwbacks to the more fish-like organisms. I'm getting a little bit ahead of myself. So let's take a look at our next slide here. Our next slide deals with Darwin. And Darwin is credited with the theory of evolution. And I really feel I feel disappointed that he gets all the credit, because there are so many people who were trying to explain the natural world around them. They were trying to explain the relatedness that they saw between species. They were trying to explain really unusual differences. They were trying to understand the world around them. And yet Darwin's is the name that sticks with all of us and that most of us associate with evolution. So since he gets all the credit, let's learn a little bit more about him. So first of all, it's important to know the time in which he lived in. So he lived in England in the 1800s. So we have the Industrial Revolution that has been occurring. We have this huge population growth. We also have colonialism. And he gets to benefit from colonialism and being at the higher levels of a really, really strict hierarchy. So his father was a doctor. And while a doctor means that you do get to be a little bit higher up in the hierarchy, similar to like what we see in modern day America with our own doctors, being a little bit higher up in the class status, you want to make sure, at least at this time in England, that you can keep that class status for your children. And so Darwin's father really, really, really wanted him to become a doctor. But he was going to have none of this. When he went to medical school, he passed out at the first operation, at the first sight of blood. And so this did not bode well. Now, never mind the fact that he was cutting classes fairly frequently and was spending most of his time really walking around the woods, making observations, and being curious about the world around him instead of being super focused on his studies. So at the time, his father was really having this dilemma of what am I going to do with this kid who's kind of a screw up? And if it wasn't for Darwin's uncle and his mentor, a botanist named Henslow, stepping in and intervening and saying, look, Darwin's he's a good kid, he's going to be okay. Let's just find something else for him to do. I think that Darwin would have had a very different future. So the mentors and his uncle intervene, and between them and his father, they get Darwin paired up with this naval ship, the Beagle. Now, the Beagle had a captain named Fitzroy, and being the captain of a Navy vessel meant that you were at a higher class status in the hierarchy. Again, we have to remember the social context here, right? if you haven't hung out or spent much time with British people, they still have a lot of this sense of the hierarchy, and they have a sense of tradition. So my academic advisor at UW was from Britain, and believe me, we all had to learn how to make tea, and you had to make it the right way, which of course is the British way. And so if you didn't, then you could suffer some severe consequences. So they still have that sense of, what you have to do in order to be at certain class levels, and this adherence to social norms. So Fitzroy, even though he's on a ship that's going to be gone from England for about 5 years, and he's going to be out at sea and in these very desolate areas, there's still this expectation that you maintain the social hierarchy, you maintain the status quo, you maintain this strict sort of structure in your life, and that includes sitting down for tea and having very intelligent conversation. And so when they bring Darwin to join this voyage, Darwin ends up being the companion to help Fitzroy to maintain this essentially social hierarchy. There was a naturalist on board whose job was to collect samples and to make observations, and that naturalist was very jealous of Darwin. There was some tension there over the course of the Beagle's journey. So here in this map we see the journey of the Beagle and the places that it would stop. And essentially what would happen is the ship would stop at port, drop Darwin off. Darwin would hire some people, go walk around in the woods, make observations, send a bunch of things back home, packages of fossils and even living samples. It's weird to think at the time that communication was fairly quick from the major ports at this time. So if they were in a port, say at the Cape of Good Hope or in Australia, it would take about 2.5 weeks to get a letter, to get some packages back home. Now the people back home were opening up these packages and going, wow, great, I love these fossils. It looks like the following things in our collection at the Natural History Museum. It looks like the following things that I saw when I was in Borneo or when I was over in New Zealand and making my own observations. And so there was this back and forth communication between Darwin and his colleagues back home as he was traveling with the Beagle, and he was making all of these observations. They were trying to piece together how everything fit together. So when he gets back to England at the end of this journey, he marries his first cousin. They have 12 children, 2 of which die. And he goes on to continue being a naturalist. He makes a lot of observations about worms and agriculture, and he contributes to a lot of other areas in science, but we don't often talk about him and his contributions to science. We focus on his theory of evolution that took him 20 years to write. He was working on this theory of evolution while he was on the Beagle, and then when he returned to England, he was talking to a lot of his colleagues, specifically Alfred Wallace, who also was working on a theory of evolution, coming to the conclusions of the fittest survive, natural selection, and a lot of those core concepts that we now associate with the theory of evolution. So why do we know Darwin more than we know anybody else? Well, part of that has to do with who he knew. He knew the publishers of a major publication, and he was able to get to them first and get his work and his ideas published first. And now his is the name that we have associated with the theory of evolution. So when we look at both his work and his colleagues' work, everybody was relying on fossils. Fossils are the mineralized remains of once living organisms. Here in this picture, we're looking at fossilized trilobites. And of course, fossils are hugely helpful. There are observations of organisms that are no longer with us. So when Darwin observes a sloth that is 6 feet tall, now think about the sloths that we have today. They're slow moving. They crawl along the ground. They don't even get up and walk like the rest of us mammals do. They only poop, I think, once a month or something. They're just extreme organisms. So Darwin discovers these bones of a 6-foot sloth. And he's going, now wait a minute, this looks a lot like this tiny little organism over here, but how in the world did he get to be 6 foot? And why is it gone today? The trilobites are an excellent example of a group of species that we don't have a lot of information on. We know that the only living ancestor that we have for the trilobites is the horseshoe crab. But other than that, we don't have much information about what they eat, how they ate, how they made their babies. Did they hunt in packs? Did they hunt alone? Were they prey? Were they predators? Were they both? What are these organisms? So even today we have fossils that we're digging up where we just are still trying to work it out. And if you get into the world of dinosaurs, and specifically the feathered dinosaurs and the connection between dinosaurs and birds, you will quickly discover, A, how important the fossil species have been in helping us understand that connection between birds and dinosaurs, and more importantly, how feathers even developed in the first place. But you will also get into an area where there's just a lot of speculation, because all we have are the mineralized remains. We don't see their behavior. We see how they were caught, how they were trapped in this rock. And so we do have to make some extrapolation. Now, it is based on logical conclusions and based on our observations of living organisms today. So it's not like we're reaching into the ether and just coming up with something. We do base it on what we are observing and what we know. But we also understand that because we can't see them alive, that we aren't 100% sure that this is exactly what they did, how they behaved, how they ate, all of the following things. So in order to understand a little bit more about a fossil and why they're so important to evolution, beyond just the morphology and what we can physically see of these organisms, we have to understand or remember a little bit about how sediment is made. Now, sediment is that collection of dirt and silt and things at the bottom of a waterway. Now, in order for those particles to collect at the bottom of a waterway, it implies that our waterway has to be kind of slow moving. So there are a lot of things that affect sediment. It starts with erosion, water falling on the landscape. When water falls on the landscape, water is going to pick up little particles of soil. It's going to erode the rock and take some of those minerals with it. So when we have erosion moving the landscape essentially into the waterways, then we have two things affecting how that occurs. First of all, how much water is falling on the land, the type of land that it's falling on, how easy it is for the water to grab a hold of those little particles, how quickly it's going to move those particles into the waterway. Once we get into the waterway, we have a couple of things affecting the formation of sediment. Faster water is going to move all particles, whether they're big or small, further through that waterway. Slower moving water is going to allow all of those particles to fall out of the waterway and to collect at the bottom a lot sooner. Our larger particles are going to collect on the bottom first. They're going to fall out of the water column and become sediment first, while those smaller particles are going to move further through the waterway and then become sediment at a later distance. Now, if you want some brutal math calculations on the rate of erosion and the rate of sediment formation, you can do that. In geology, they have figured all of this out. They know the rate of sediment formation for just about everywhere on the planet. And so we're very, very confident in how quickly these layers will accumulate in different bodies of water. That then allows us to understand how quickly those layers accumulated in past bodies of water. So here we're looking at the Grand Canyon. And what I love about the Grand Canyon is we see that sedimentary rock. We see all of those very different layers. And they're distinct. I mean, in this picture, we see that light tan band, and then we see this really sort of deep red. And the Colorado River has been cutting through this so that we can see into those different layers. I have to encourage you, if you haven't done so already, to get down to the Grand Canyon and not just stand at the top and stare over the edge and go, wow, and feel that depth and feel the distance of the Grand Canyon, but I do encourage you to hike down a bit. Because in hiking down through those layers, you're going to get different smells. You're going to get different feelings. Some of the rocks are going to hold heat better than others, so you'll kind of walk through these layers that are warmer and some that are cooler. Even on a blazing hot day, it's really, really, really remarkable to descend into the Grand Canyon. Now, the hiker in me always wants to tell you, be extremely careful if you do this. Plan your trip. Don't just show up at the Grand Canyon and be like, yeah, man, let's hike to the bottom. Because that's how people die every year. Even the most fit and athletic people, on a spur of a moment, decide that they're going to hike to the bottom and back again. And it's a much more arduous trip than that. You really do need to put in some planning. So all national park people will warn you. There's signs everywhere that say, please don't. Just make sure you know what you're doing. Don't do this on the spur of a moment. Because it is a very, very, very deep, very intense experience. But what the sedimentary rock has allowed us to do, this and other sedimentary rock from all around the world, is to develop the law of superposition. So based on that rate of sediment accumulation, we know that any animals that are trapped further down or deeper in the sediment are going to be older, and that those nearest to the top, like say the surface of the Grand Canyon, are going to be a lot younger. This gives us an approximate age of those organisms when they would have lived on this planet. But an approximate age doesn't feel real good. We'd like more detail and we'd like more accuracy. And this is where we get into the role of radioisotope dating, radiocarbon dating. So when we have an isotope is going to have more neutrons in its nucleus. It has a predictable rate of decay. In other words, how long is it going to take for that atom to transition and become something else? This is where we get to the concept of half-life. So we can calculate, based on what we have in front of us today, how long it would have taken that isotope to decay into the form that we see at this very moment. So our fossils are going to have those isotopes, and those isotopes are going to help us to calculate a more accurate time range when that organism lived on our planet. So our radiometric dating is going to be using isotopes to age and date an organism. Right now, you, as you are living, are storing little isotopes in your body. So if you become a fossil someday, somebody could use your isotopes to figure out that you were in existence right now on the planet during this time frame. They probably narrowed down to the year 2000 to 2000, and let's hope you live this long, 50. So we have two ways that we age our fossils. The first is through that law of superposition, and then we have radiometric dating. So once we have these different fossils from all of these different species, we're going to be using that morphology, those physical characteristics, to develop what's called a phylogenetic tree. A phylogenetic tree is just an abstract tool showing the relatedness between species. And if all we have is our fossils, then we're going to use just the morphology. So in our example of Tiktaalik, here we have several different species. There's a lot more species in our phylogenetic tree than what's being shown here. And we're tracking a couple of main characteristics. In blue, we see this kind of bony shield at the base of the skull. And we can see where that was lost. In other words, when Tiktaalik speciation event occurs, and we end up with Tiktaalik individuals, they no longer have that bony shield at the back of their skull. So that would be an ancestral characteristic that then is lost in Tiktaalik. But then if we look at that green set of structures on the skull, we can see that that characteristic sticks around, even through further organisms or further speciation events on the planet. We get some modifications, but that structure sticks around. So that too would be an ancestral characteristic that keeps on getting passed on. So what's the importance here about supposed missing link fossils? Is that they can help fill in the gaps of when structures disappear, like that blue bony shield on the skull, and when those structures, or why those structures, or how those structures keep sticking around even when speciation events keep occurring. So we're looking at another phylogenetic tree here, and in this case we're expanding it out a little bit longer. We can still see Tiktaalik connected all the way over to our modern day frogs, but we see that connection over to fish like the coelacanth. And the coelacanth is one of those really unusual fishes. It has hip bones, like our hip bones, and it's got hind limbs, kind of like our legs. And when it swims almost like it's walking. Its legs, its fins move in this kind of opposite side motion. It's very unusual compared to other fish. And the bones that we see in coelacanth are modified, and we see a lot of those bones in Tiktaalik as well. So expanding our view to look for those ancestral and derived characteristics, to see how organisms are connected, to see that change in the line of descent over time to the organisms that we now see on our planet today. So when we're doing this, we're looking for common ancestry, and we're looking for homology, that similarity due to common ancestry. So there is this misconception in evolution that when we have speciation, that all of a sudden these new things develop out of nowhere. And every single time what we have discovered is that that's not the case. It may look new to our eyes, but when we start to understand the structure, we see that's just a modification of what came before. In other words, when we have a speciation event, it can only be based on everything that has come before. Things do not spontaneously arise in evolution. So here we're looking at homology from the DNA point of view. We can get a hold of DNA. We look for the similarity in those letters. This reinforces what we see with the morphology, those physical characteristics. We're looking for the similarities and the changes, that shared ancestry. So when we didn't have DNA, we were forced to focus on anatomical evidence. So one thing scientists would look for is they would look for homologous structures. Homologous structures are going to solve different problems. They're going to serve different functions for different species, but they're going to have similar components. So remember, our prefix homo means same. In this case, the sameness is going to refer to the bones, the components of the thing that's doing whatever that organism needs it to do. So a classic example is looking at mammal forelimbs. When we look at mammal forelimbs, there's not a whole lot of creativity here. Almost all mammals have exactly the same forelimbs. A forelimb consists of a humerus, a radius and ulna in orange and tan, carpals in yellow, metacarpals in green, and phalanges in blue. Now humans are going to use their forelimb for grasping and grabbing things, while a cat is going to walk on its itty-bitty little phalanges. It's going to be able to use its forelimb for running and grasping and catching some prey, but pretty brutally when it does that. A whale has the same humerus, radius, and ulna. Granted, they're all short, but they're all still there. Carpals, metacarpals, and elongated phalanges. And those elongated phalanges are essentially going to help our whale to be able to swim. And finally, our bat, which is the other extreme example here of what to do with a forelimb, still has a humerus, radius, ulna, carpals, elongated metacarpals, and phalanges. And with that skin stretched between the phalanges, the bat is able to fly. So we see the same bones, the same components, doing different things for different species. Now here in this picture, it's the same thing. We see forelimbs, we see homologous structures. But what I like about this is the horse's forelimb, because the horse's forelimb is so incredibly extreme. If we see the humerus, it's similar to the humans. Our radius and ulna in blue and yellow. Our carpals and metacarpals are in green, and our phalanges are left uncolored. And if you notice, the horse, what we would call the leg, commonly called the leg, is really actually one great big long middle finger. So a horse is just running on its middle finger. And when you think about the speed and the force and the pressure that one finger is taking, it is remarkable. It is absolutely amazing, because our middle fingers can't take that much pressure, can't take that much force, and definitely can't be used to run or jump or carry our body weight. So it's the extreme example of modifying what came before. Previous species of horses, before we get to the modern horse speciation event that we have today, did have all 5 of the phalanges, But what we saw over time was a preferential use of that middle phalange, and the other fingers essentially fell off and gave rise to the extreme example of horses that we have today. Another thing that we'll be looking for is we'll be looking for analogous structures. Now again, analogous structures are used to solve the same problem. They serve the same function for different species, but are made of different components. So when we look at flight, there are a lot of different wing-like structures that can be used for flight. On the top we see a chicken wing. When we look at all bird wings, the bones are sort of similar to mammals. There are some significant differences that you have to get into ornithology to really understand. I think the bigger point here is the idea that our birds are going to be using feathers, and those feathers are going to be incredibly wonderful for flight. They can actually move and tweak each individual feather. They've got muscles attached to them, so they can have this precision in flying. Now, a lot of birds can migrate. They can migrate big, long distances. Bats can migrate as well. We've got a bat wing down here on the bottom, but the bats are using skin, and that skin is not going to give them the same level of precision that birds with feathers do. So analogous structures, they both achieve the same goal of flying, both allow the animal to fly for long distances, but very, very different reason why they are able to fly for long distances. Feathers versus skin. The final thing that we'll look for are these vestigial structures. These are structures that stick around after speciation has occurred. And while they may have a function for one species, maybe a distantly related species, they don't have a function, or they have such a diminished function that they might as well not exist for another species. Now what's interesting is that the cells are still reading and expressing those genes. And if you think back to chapter 11, and all of the stress and strain of transcription and translation, gene expression to make those proteins, the cells are still doing all of this, even though that structure doesn't have an obvious purpose. So the unanswered question that we have in evolution and in cellular biology is, why are the cells still reading and expressing these genes? Why aren't those genes turned off yet? It doesn't make sense to invest the energy if you're not going to get some direct gain out of it. So a classic example is the coccyx bone in the humans, and tails in other mammals. Tails in other mammals allow them to be able to balance, while our coccyx bone has no real function except for to break whenever we fall on the ice. And the other example are ostrich wings. In the picture here, we have the ostrich wing that doesn't serve a good function that we know of, but there is a tremendous amount of energy being invested in the feathers. That's a lot of keratin protein being used for, well, we don't know. It certainly doesn't allow them to be able to fly. And on the bottom, we have a pairing of a lemur and the human coccyx tailbone to show how diminished that tail structure is in our species of mammal. Another interesting example comes with our aquatic mammals, where up until a certain point in the embryonic development, they're making a hind limb. They're reading and expressing those genes circled to make that hind limb circled here in red, and that after about 9 weeks of development, those genes do turn off, and the body will reabsorb those cells and reabsorb the structure back into the body. So they're still making that hind limb reminiscent of life on land. So morphology, like homologous and analogous and vestigial structures, are used to show the similarity and differences between species. So the whole foundation, or the big theme in evolution, is the biological species concept. This idea that members of a species interbreed and make viable offspring. They can keep perpetuating the species. They have a shared gene pool. All of those genes that are possible for any individual in that species. Species are reproductively isolated from each other. In other words, they're not going to interbreed and make offspring. Our species are made up of populations, and species have common ancestors. Now, when we look at speciation events, sometimes we get really significant differences. So here we have a picture of a robin, and it's hard to believe that the robin and the bluebird are closely related to each other. They look so different from one another, but they are closely related to one another. And we have this point where speciation occurred, the splitting, and it gave us really obvious differences. But sometimes we get really, really subtle differences, and in the flycatcher family, it is absolutely challenging to try and tell the difference between different species. So we have the willow flycatcher here on the left and the alder flycatcher on the right. And having handled hundreds of these birds, I can tell you that even with a trained eye, it's incredibly challenging to tell the difference. You really can't until they get onto their breeding ground, and then they make different songs, and that's the only way that you're going to be able to tell these two species apart. So speciation can definitely be very, very, very confusing. We hope it's always really dramatic, like the robin and the bluebird, but sometimes it's incredibly subtle, and that can make it harder to see changes in a line of descent happening. Now, we total up everything that we know, from the DNA to the RNA. We're going to plot our understanding of how species are related to one another on something called a phylogenetic tree. And Darwin started working this out. He was starting to think about how species give rise to new species, the similarities, those ancestral traits, the derived characteristics. We now have much more sophisticated tools to put together these phylogenetic trees. So it's not meant to tell you exactly when speciation occurred, but it's meant to plot these changes over time to help us to understand how species are related to one another. So this concept of phylogenetic trees is going to launch us a little bit into chapter 16. But since we're already talking about speciation, for me it made sense to pull these concepts in here. So here we're looking at a phylogenetic tree. We have our species just labeled abstractly as A, B, C, D, E, F, and they're out at the end of our branches. What I am showing here is a relatedness between these species A, B, C, D, E, and F. Circled in red, I'm going to have a node. Where one branch connects to another, what I'm communicating is that I have an evolutionary event. Some kind of change in a gene or multiple genes, something happened. And now species C is no longer able to interbreed with species B or species D. I have that separation in species. At the very end, I'm going to have this root, and that's connecting all of these species that I want to look at back to the universal tree of life with the 8.7 million named species. In other words, I always want to communicate that I'm zooming in on just a very small portion of life on this planet, and I know that these individuals are connected to everything else that we have learned about and discovered on this planet. So when we read a phylogenetic tree, what we're going to say is that species E and F are closely related to each other because they share one node in common. Species F and species A are distantly related because look at how many nodes, how many speciation events have happened separating species F and species A. If we swap species E and F here at the end, we're still communicating the same information because they're still sharing that same node. But if we swap species F and species B, now we have rearranged the evolutionary events. Now we're communicating a very, very different relationship between the species on this phylogenetic tree. Now there are lots of different styles to communicate this information. All four of these phylogenetic trees communicate the same information. They're all saying species C and D are closely related to each other. They all share the same node, and they're all distantly related. Species C and D are distantly related from species A. But we have preferences to the styles that we like. I don't like some styles. You'll find which styles work for you. Now, there's another tool that we use in evolution to communicate the relatedness between species, and this is cladistics. And you're going to see cladistics more in education. And the reason why is because cladistics relies on morphology. We're not trying to communicate as subtle details. We're not looking for refined relationships a lot of times with cladistics. We're just going to focus on morphology. And so here we're looking at a data table of morphology. We've got groups of organisms, lancelets, eels, newts, snakes, and lizards. And hopefully when you look at this, the first thing that jumps out at you is the idea that you know that there are hundreds, thousands of species, different species of lizards. It's not just one species. So we're comparing these broad categories of lizard, broad category of snake, broad category of newt. But we're zooming in on just a few morphological characteristics. So what we want to see is who has a notochord in the embryo, who has vertebrae, who has lungs, when did the three-chambered heart develop? How about internal fertilization and that amniotic membrane in the egg? What about four bony limbs and a long cylindrical body? Which of these morphological characteristics do these groups of species share in common? And when do those characteristics disappear? Or when do they appear? When do we have derived characteristics, and what do we have as an ancestral characteristic? So looking at the top of our list, shaded in blue, we see that our ancestral characteristic is the notochord in the embryo. All lancelets, all eels, newts, snakes, and lizards have this characteristic. But we see a speciation event where the eels develop vertebrae. So that would be a derived characteristic. Once it shows up in the eels, all the newts, all the snakes, all the lizards, everybody thereafter, every species that is new to our planet after that will have the vertebrae. Same thing with lungs and the three-chambered heart. Our newts are the first to show this, but once our newts develop that, every species after that is going to have lungs and a three-chambered heart. Our snakes are the first to develop internal fertilization and the amniotic membrane in the egg. Once snakes as a species have this characteristic, this derived characteristic, they will all have that from that point forward, as will the lizards. Now, when it comes to four bony limbs and a long cylindrical body, these morphological characteristics aren't going to help us in determining derived versus ancestral characteristics. It's because some species have it, others don't. And so, in general, when we move from that data table to plotting it onto a phylogenetic tree and showing the relatedness over time, we're going to eliminate the characteristics that don't show us a clear tracking of ancestral characteristics and derived characteristics with speciation events. Now, these phylogenetic trees are really helpful. So here we're looking at how the gray wolf is connected to a lot of our domesticated dogs, and it's also showing us how some of our domesticated dog breeds are connected to one another. This could help us to understand behavior. It could help us to understand our own individual pet that we have living with us, maybe health problems. I don't find that this circular phylogenetic tree works for my eyes. It's really, really hard for me to see how the scent hounds are closely related to the spaniels, and then to track how they're then, if I have to follow all of the lines, back to the gray wolf. It's just confusing for my eyes to follow. I really like the rectangular or the kind of triangular phylogenetic tree that I showed you in the example. I think that that's easier on my eyes. And then, since I know that there are some cat fans out there, I didn't want to ignore the fact that we have phylogenetic trees for cats, showing how some of our kind of iconic breeds, like the Norwegian forest cat, the Maine Coons, the Persians, the naked cats, are all related to one another. Again, looking for shared traits, derived characteristics, and ancestral characteristics, and potentially disease situations that we may not want in future cat pets. So phylogenetic trees are very helpful. You'll see them a lot in evolutionary biology. It's the fastest way to communicate a lot of information and a lot of detail in a way that most people can very quickly get and intuit. So we don't have to go into a whole lot of explanation with these phylogenetic trees, which is why we love them in the field of evolutionary biology. So this brings us back to our phylogenetic tree of life, where we have bacteria, archaea, and eukarya, our three broad domains. And then within those domains, we break them into kingdoms. We get down to the genus and the species level. And if we were to look at all 8.7 million named species on this planet, we would have this very complicated phylogenetic tree, this universal tree of life. If you notice in the upper left-hand corner, we've got the kingdom of animals and some little writing that you can't read that says you are here, and that's showing how Homo sapiens, our genus and species, is connected to the rest of the life on this planet, our relatedness to all of the other named species on our planet. Now, before we leave chapter 14 and this big overview of evolution, I want to talk a little bit about convergent evolution. Convergent evolution is very interesting because what we have is we have two distantly related species, often living in different geographic locations, looking very similar to one another. In other words, these species are converging on similar solutions to similar problems in the environment, which is why they look so similar to one another. But the mistake that we can make is saying that these individuals are closely related to one another, when in reality they're not. So euphorbia will only grow in Africa, while cacti will only grow in the Americas. But here on this picture, we can see that these two species look very close to one another. Our knee-jerk reaction is to say, well, they must be related to each other. Look at them. They look like one another. But the reality is that they are distantly related. They would never be able to interbreed. But they have some characteristics that allow them to survive in desert environments. They have segmentation that allows them to swell when there's rain and contract when there's no rain and not break apart. They've got spines for protection, and they've got these great big chunky stems for doing photosynthesis. We see convergent evolution in mammals as well. The flying squirrel lives here in North America, has flaps of skin between its appendages for gliding from tree to tree, great big eyes for seeing at night. This is a nocturnal organism. It even has coloration that allows it to hide from predators. It's brown on top so it will blend in, and then when it's flying through or gliding from tree to tree, it has that kind of light underbelly so it blends in more with the sky. Again, if you're looking up, it's hard to see it if you're a predator. If you're looking down, it's hard to see it if you are a predator. The sugar glider is a marsupial. It lives in Australia, and we see a lot of those similar characteristics, again converging on similar solutions to similar problems. These are both small mammals that everybody wants to eat. So how are you going to make sure that you can get food, you can make your babies, and not get eaten? And so what we see is they are converging on similar solutions. Now, divergent evolution is where we have distantly related organisms, often living in the same geographic location, end up looking very different from one another. They often have homologous structures. So for a long time, the hyena were classified as dogs. They look very doggy. They've got that elongated nose. They've got the kind of German shepherd slope to the back. They hunt in packs. They have a strict social hierarchy. They even kind of make a yippy barking noise. Everything about them screams dog. But they're actually more closely related to cats. And they're more closely related to the African wildcat, which, ... is closely related to our domesticated cat. But look at that morphology. It would be hard to say that a hyena looks like that African wildcat, that they are closely related. Yet when we get a hold of the DNA, what we see is that there are a tremendous amount of similarities. So here we have the African wildcat and the hyena competing for resources, diverging from one another. They're using their bones, they're using their similar genes in very different ways in order to outcompete one another and survive in their environments. So that's a very big overview of evolution. The next two chapters should be a little bit shorter and a little bit faster. I wanted to leave you with this quote from The End of Darwin's Life, because we focus so much on the strongest surviving. And what he realized, and what many other of his colleagues realized, and what researchers today realize, is that it's not necessarily the strongest that survives. It's not even necessarily the most intelligent. It is the individuals that are the most adaptable to change. The environment is constantly changing. Those that can adapt to the changes, those will survive. And that is ringing so true when our world is changing so quickly. And we see those species that are going extinct, and we know the ones that will survive. Unfortunately, they're the ones we probably don't want to be around. Things like cockroaches and mice and raccoons. They've learned how to live with us humans. They're learning how to live in our human system. Those that we often love may not be able to adapt to the changes. If you have any questions, email me, drop into Zoom office hours.